Today we'll be summarizing the information found in Module 10, Evolution. Please locate your graphic organizer, which can be found at the end of Module 10 in your Biology Student Handbook. We're going to begin by going over the sequence of events as it pertains to the origin of life on Earth. So on your graphic organizer, you will see several little panes where you will be drawing simple pictures. The picture that you need to draw for the first pane is shown on this slide. You have what resembles a bowl containing amino acids. You have a lightning strike to represent energy hitting that bowl. And you have chemical formulas floating around that bowl to represent gases in the atmosphere. CH4 is methane, H2S is hydrogen sulfide, N2 is nitrogen gas, CO2 is carbon dioxide, and CO is carbon monoxide. This diagram represents abiogenesis. Abiogenesis means without life beginning, if you literally translate it. You have a box on your graphic organizer just beneath the word abiogenesis in which to record some information. You want to record that abiogenesis began when life first arose from non-living material in what we call a primordial soup. And this is why we have shown a bowl in the image. We have confirmation that this could have occurred when the Earth was young because it was experimentally tested by scientists Miller and Urey. They confirmed in a laboratory setting that with this combination of atmospheric gases and intense energy from lightning strikes or volcanic eruptions, amino acids, which are the building blocks of protein, could arise where there was no organic material previously. Because this represents life arising from non-living material, we also call this process spontaneous generation. Looking at the second step in the sequence, you have a cell to draw which resembles a little Pac-Man. He is eating some molecules, and you will also notice that there is no oxygen in the atmosphere. So you need to make sure that you record all of that information in the pane for your drawing. This is the first step in the evolution of cells. And this evolution of cells is called the Heterotroph Hypothesis. And you will notice that beneath the next three panes on your graphic organizer, they are bracketed to indicate that each of these is a step explaining the Heterotroph Hypothesis. There is a large box beneath the term Heterotroph Hypothesis in which you are going to write some information. The information that you first need to write in that box is that the Heterotroph Hypothesis explains the evolution of prokaryotic cells. And of course, you remember prokaryotic cells are those without a nucleus. That's all you need to write in that box right now. If you look up underneath that pane where you drew this picture, you will see that it's already labeled with some terms. These cells were prokaryotic, the simplest type of cell. They were anaerobic, meaning that they did not require oxygen. And they were heterotrophic, meaning that they had to take in nutrients from their environment. The second step in the Heterotroph Hypothesis is shown by drawing a cell releasing oxygen when the sun is present. So make sure you draw that picture in the pane provided. You will also Note underneath that picture that these are still prokaryotic cells with no nucleus, but because they are using the process of photosynthesis, they are releasing oxygen. This is what led to the formation of the ozone layer, which protects life on Earth from ultraviolet radiation and allows life on Earth to move out of the water and onto land. The third step in the heterotroph hypothesis should be shown by drawing a cell taking in oxygen from the atmosphere. So draw this picture in the pane provided. You will notice that this is still a prokaryotic cell, but it is an aerobic cell because it is using oxygen. Now go back down to the box labeled Heterotroph Hypothesis. You've already written that this explains the evolution of prokaryotic cells. Next, you need to write that it is called the Heterotroph Hypothesis because the cells start with heterotrophic cells taking in nutrients from their environment. Next, we move on to the Endosymbiont Hypothesis. Please carefully draw the picture shown on the slide in the space provided. It begins with a cell resembling a Pac-Man who is taking in two much smaller cells. You will notice that one of those cells is colored a red color and the other is colored a green color. The second stage of the picture shows those two smaller cells within the larger cell. Now you have a place to explain the endosymbiont hypothesis. Locate the box below the word endosymbiont hypothesis. Please record that this explains the evolution of eukaryotic cells. Also record this explanation. A symbiotic relationship formed between larger and smaller prokaryotic cells. You can see this in the picture. The smaller cells eventually became what we now know as mitochondria and chloroplasts. The last step in the process of the evolution of life on Earth is the evolution of multicellular life. In each of the images that you have drawn previously, you have been drawing single cells, unicellular organisms. These unicellular organisms started out simply as prokaryotic cells, evolved into more complex eukaryotic cells, and from those eukaryotic cells, we have the evolution of multicellular life. So please draw a picture of a flower in the last pane. Please also write the following information in the box beneath the words multicellular life. Multicellular organisms are made of eukaryotic cells, and the evolution of eukaryotic cells allowed multicellular life to evolve and eventually colonize or live on land. There is nowhere to record this on your graphic organizer, but please keep in mind that while abiogenesis means that life arose spontaneously and that this is how life probably began on Earth, we now know that life continues through other life. This is the theory of biogenesis, life beginning. So although abiogenesis may have occurred when the Earth was young and the atmosphere was different, it does not occur today. Spontaneous generation for life today has been disproven by scientists. Look down at the bottom of your graphic organizer and you will see this picture. you need to write in that this was the experiment that was done by Francesco Redi, which disproves the theory of spontaneous generation. As a reminder, Redi had two containers, both of which contained rotting meat. One of them he covered, the other he left open. He noticed that the flies entered the open container, but could not enter the closed container. Over a period of several days, maggots or baby flies appeared on the meat in the open container, but not on the meat in the closed container. He disproved the idea that the maggots came from the meat. He proved the idea that the maggots came from the flies. The next picture shows an experiment done by Louis Pasteur, and you need to record his name. He disproved spontaneous generation at the microscopic level. Here is how he did that. He took chicken broth, which contained microorganisms in it. He knew this by looking at the broth under a microscope. He put the broth into a flask called an S flask due to the shape of its neck. He boiled the broth to kill the microorganisms and confirmed that they were killed and that the broth was clear. He left that flask for a long time, and over a period of a year he checked that flask periodically and confirmed that there were no microorganisms in the flask. However, when he broke off the neck of the flask, he noticed that microorganisms began to grow in the broth. He proved that microorganisms could get into the flask when the neck was broken off and they reproduced. But when the flask had its neck intact, the microorganisms were being trapped at the low point of the neck and could not get into the broth. Therefore, the broth did not lead to microorganisms spontaneously. He disproved that idea. Instead, the microorganisms arose from other microorganisms. On the back side of your graphic organizer, we are going to look in depth at the process of microevolution, which is the most important theory for you to learn and understand. Please, in the space provided, write that evolution simply means change over time. Evolution occurs through the process of natural selection. If you look on the left side of your graphic organizer, you will see the statement, how does evolution occur, and natural selection, with a space to write some information. Please write in that space that this means nature selects the best adapted organisms to survive and reproduce. So what is an adaptation that makes an organism better suited to its environment? What makes an organism the best adapted for a particular environment? If you look again under the area for natural selection, you will see a space to write some notes about adaptation. Please record that adaptations are heritable traits. These are traits that can be passed from parent to offspring. Also record that adaptations make an organism better suited to its role in the ecosystem. And finally, record that adaptations may be structural, behavioral, or physiological. An example of a structural adaptation might be the sharp teeth of a lion. The lion is adapted for tearing into meat. You can see some other examples of structural adaptations on the slide: camouflage, For example, when an insect blends in with its environment or a frog blends in with its environment. Mimicry, when one organism resembles another organism. These are examples of structural adaptations. Behavioral adaptations are those like herding or schooling or growling, things that organisms do that make them better suited to survive and reproduce. And finally, physiological adaptations are those things that organisms have that you can't necessarily see, like specific enzymes to drive their metabolism, their various types of ability to transport oxygen throughout their bodies, the capabilities that they have for sight, for example, the compound eye of an insect. All of these are physiological adaptations. We're still continuing down the left side of your graphic organizer. The theory of natural selection was proposed by Charles Darwin. This is an important scientist to remember, so please record his name. There are 4 requirements for natural selection. You're going to write these 4 requirements in and briefly explain each. The first requirement is variation in a population. This means there are different phenotypes or physical traits within a population of organisms. So looking at the picture, you will see that in a population of peppered moths, which is a specific type of moth, there are some that are completely dark, there are some that are completely light, and there are some that are a mixture of light and dark. The next requirement, record this under number 2, is overproduction. In a population, there are more individuals produced than can survive. This leads to competition in that population. So on the slide, you will see a fair number of moth eggs that have been laid. Not all of these moths are going to grow to adulthood because they are going to have to compete for the resources they need. The third requirement for natural selection is natural selection. This simply means nature selects the best adapted organism. This is best explained by again looking at the picture. If you look at this picture, you will see that one of those moths is better camouflaged on the dark colored bark. Nature selects that moth to survive. He is the best adapted in that scenario. The lighter colored moth is going to be more easily seen and is going to be eaten by a predator. The last requirement for natural selection is survival of the fittest. This is actually better described as survival of the best adapted individuals because they have better reproductive success. So let's look at the last picture. This is an actual scenario that occurred around the turn of the 20th century in England when industrialization became very heavy. The light-colored bark of the trees in the forests around England was able to camouflage the lighter-colored moths before the Industrial Revolution took off. So as you can see on the first half of the picture, most of the moths in that area were light in color. But during the Industrial Revolution, soot from the factories coated the bark of the trees, making them darker. Over time, the lighter colored moths were more easily seen by predators and were eaten more frequently by predators. The dark moths survived and reproduced, passing their dark-colored genes to their offspring, leading to a greater population of dark-colored moths over time. Looking now at the middle of your graphic organizer, it's very important that you understand that a single individual does not evolve. You're going to fill in the blank. Individuals don't evolve, populations do. This requires a change in the gene pool of the population. Fill in the blank on your graphic organizer. Now let's look at the picture shown on the slide. We have a population of wild boars. Some of them have the genotype for a particular trait, big B, big B. Others have the genotype big B, little b, and others have the genotype little b, little b. Each of them expresses either the dominant or recessive version of the trait. Big B, and big B, little b represent the dominant version of the trait. Little b are going to express the recessive version of the trait. Collectively, all of these alleles, big B's and little b's, represent the gene pool of this population that is living together, interacting, and breeding. Going back to your graphic organizer, mutation is the raw material for a change in the gene pool. Sometimes a mutation is good, and it can be selected for. Organisms who have that mutation would be at an advantage. Sometimes a mutation is not good. It can be selected against. Individuals with that mutation would not have an advantage. Make sure you filled in the blanks on your graphic organizer. Now, when natural selection occurs, it can take place in 3 different mechanisms. These 3 mechanisms are illustrated for you on your graphic organizer. So please carefully look at the diagrams and make sure you understand the explanation. The first mechanism is stabilizing selection. So write that in the first blank beneath 3 selection mechanisms. Now look at the 2 graphs just beneath the word stabilizing. The first graph represents a normal distribution curve, where some individuals are at the low end, most of them are in the middle, and some are at the high end. After stabilizing selection, the average form of the trait, the average phenotype, is selected for. So the curve actually gets more and more average. There are fewer individuals at either end of the curve. The second selection mechanism is directional selection. Again, we start with the normal distribution curve. But in this case, individuals at one extreme are at an advantage. So over time, looking at the graph beneath the first, the curve moves over. In this case, the curve moved to the right. It's more favorable to have the phenotype at that extreme. For example, it might be more favorable for a woodpecker to have a larger, longer bill because it can get to more insects beneath the tree bark. The final type of selection mechanism is disruptive selection. So again, we have the normal distribution curve. Over time, both extremes are selected for, and the average is selected against. So over time, there are more individuals at either extreme, but fewer in the middle. For example, you might have clams that are white, gray, and black, and the white ones would be selected for, as would the black ones, but the gray ones would be selected against. As a result of natural selection, sometimes individuals in a population become so different that they actually form a new species. You have a spot on your graphic organizer to record the definition for speciation, the development of a new species. So let's go ahead and write down the sequence of events that occurs. It begins with geographic isolation, meaning that a population that was once living together and interacting and breeding together, some of the individuals are separated from the others. In the picture, you can see a canyon that has formed, separating the population of pine trees. The second step is natural selection. Nature selects the individuals that are best adapted for the particular environment. So in the picture of the pine trees, there might be some climatic condition on one side of the canyon that is different from the climatic condition on the other side of the canyon. So over time, different mutations in the trees are selected for, and over time, they become different enough that they are different species. That brings us to the third step, reproductive isolation. With reproductive isolation, those two groups are now so different that they can no longer interbreed. They are not able to mate and produce fertile offspring. They are separate species. They are reproductively isolated. Sometimes this process of speciation happens very gradually over a long period of time with the accumulation of small changes. If you follow the arrow over from speciation, you will see this same picture on your graphic organizer. And the picture at the top, which looks like a branching tree, should be labeled gradualism. Down at the bottom, we have the ancestral species, which over time changed a little bit at a time, leading to the formation of many different species. And for example, we can see how horses have changed over time with the very small accumulation of changes. And now we have not only what we call horses, but we have things like zebras and other species that resemble horses as well. The other time frame for evolution should be labeled punctuated equilibrium. This occurs with speciation in rapid bursts. So there are thousands of years of stability and then a rapid change. This is what happened with elephants. Instead of seeing the gradual accumulation of changes that we saw with horses, we have in the fossil record the woolly mammoth, and then bam, the African elephant and the Asian elephant. There were no gradual changes that were observable in our fossil record, meaning that the changes took place in more rapid bursts. Look down at the bottom of your graphic organizer and we'll label some of the pieces of evidence that we have to support evolution or the change in species over time. The first picture should be labeled fossil record. Now this picture shows relative dating of fossils. The fossils that are at the bottom, organism A, are going to be the oldest because they were laid down first. And the fossils at the top, organism C, are the youngest because they were laid down last. There are methods that can give us exact ages of fossils. This just gives us their relative ages. We know that A is older than B, which is older than C. The next picture should be labeled biochemical evidence. This is the best evidence for evolution. Remember that organisms that are similar have similar DNA. This chart shows us amino acid sequences. And if you will recall, amino acids are coded for by the DNA. Therefore, if the amino acids are similar, the DNA is similar. So look at the human, the amino acid sequence, lysine, glutamine, histidine, and isoleucine, which is exactly the same as the sequence for the chimpanzee. This gives us an indication that humans are more closely related to chimpanzees than they are to gorillas, where there is one difference, or horses and zebras. Now compare the horses and zebras, and you will see that they are very closely related to one another, with only one difference. The next picture should be labeled homologous structures. Homologous structures are those that have a similar bone arrangement, even though they may have different functions. So looking at the human arm, the whale flipper, the dog leg, and the bat wing, it starts at the top with one larger bone, splits into two smaller bones, and then splits again into 5 smaller bones. So even though the human arm is for manipulating objects, the whale flipper is for swimming, the dog leg is for walking, and the bat wing is for flying, we have some sort of a relationship between these organisms due to the similarities in their bone structure. Please remember that analogous structures, for example, the wing of a bird and the wing of a butterfly, do not give us evidence of evolution from a common ancestor. They are very different structurally, but are just adapted for a similar environment. The next picture should be labeled vestigial structures. Vestigial structures are those that are not functional in present-day organisms, but may have been important in an ancestor. So in your picture on your graphic organizer, you see the human tailbone. Obviously, we don't use a tail. Look at the picture on the slide of the snake. At the base of the snake's tail, you can see 2 spurs, indicating that at some point there may have been legs. The last picture that gives us evidence for evolution should be labeled comparative embryology. This involves comparing the stages of development in different organisms. And those embryos that are more similar have a closer relationship. Now, if you look at the picture, you see that the very earliest stage for the fish, the turtle, the salamander, and the human all look very similar to each other. But moving to the second stage of development, the fish and the salamander look quite different from the turtle and the human. And by the third stage, there are very noticeable differences. This gives us evidence that the fish and the salamander have a closer relationship to each other than they do to the turtle or the human. Once again, please recall that all of this type of evidence for evolution supports change over time from a common ancestor. In fact, go ahead and write that in beside the words evidence for evolution. Change over time from a common ancestor. People tend to mistakenly believe that evolution was something that happened a long time ago. Evolution, or change over time, is still happening. However, it's usually too slow to observe. But there are a couple of cases in which we can observe evolution. You have a place to write these examples in the top right corner of your graphic organizer. The first example is with pesticide resistance in insects. Please listen carefully. Look at the first picture. We have some pests. Farmers don't want pests on their crops. The crops are being grown for people, not for pests. So the farmer sprays a pesticide. That pesticide, looking at the second picture, kills most of the bugs, but a few are left because they had resistance. They didn't become resistant. They didn't become immune. It was already there in their genes. Now look at the third picture, bottom left. You can see that those insects that were left began to reproduce. The farmer sprays a pesticide again. This time, more of the ones that are shaded in white die. The ones in red are the ones that are resistant. So they survive and reproduce. Next time when the farmer sprays the pesticide, very few of the insects will be killed because the population has evolved resistance. Once again, one bug did not become resistant. It did not become immune. The population changed over time because some individuals had resistance to start with. The second example to record for evolution today is antibiotic resistance. Look at the picture. Let's say that you're sick with an ear infection. The doctor gives you an antibiotic. You begin taking that antibiotic. Picture 1 shows all of the bacteria that are making your ear hurt. And when you take the antibiotic, look at picture 2, most of those bacteria die. But the orange one was resistant. It didn't become resistant. It was resistant to start with. So that orange bacteria begins to survive and reproduce. Over time, you keep taking that antibiotic because you have to take it for 10 days or so. And the ones that don't have resistance continue to die. But the one that initially survived has reproduced so much, looking at the fourth step, that now the entire population is resistant and your ear still hurts. This is why penicillin doesn't work for most infections today. Bacteria have evolved resistance.